Hello, dear viewers. In this video, we will explore the principles of scaling applied from classical physics to neuroscience, specifically how theoretical frameworks rescaled from thermodynamics, special relativity, and general relativity provide insights into neural dynamics, action potential propagation, and the emergence of consciousness. Scaling is a transformative concept in theoretical physics, allowing frameworks initially developed for specific physical phenomena to be generalized to solve similar problems at different scales. In this context, we will derive symbolic representations of neural geodesics in spacetime and extend them to a proposed five-dimensional, 5D manifold, presenting a holographic view of consciousness as an emergent phenomenon. Scaling mechanisms will be illustrated using basic examples from thermodynamics and heat theory, and their implications will extend to medical imaging applications such as diffusion MRI and intravoxel incoherent motion, IVIM. Theoretical physics often relies on scaling to extend frameworks to different domains. This application was famously used in Einstein's work linking diffusion and Brownian motion. In neuroscience, by minimizing concepts from relativistic frameworks, a connected spacetime is hypothesized to have four nested dimensions shaped by neural activity. This concept is analogous to the curvature of spacetime by gravitational fields and suggests a fifth dimension that could represent consciousness as an emergent holographic property. Scaling allows theories such as quantum mechanics, QM, and general relativity, GR, to be adapted beyond their traditional boundaries. GR, which is usually applied at macroscopic scales, describes cosmological phenomena, while QM describes atomic and subatomic interactions. In the case of the human brain, which fits neither quantum nor relativistic scales, scaling offers a way to understand neural function and consciousness from a physical perspective. We will begin by outlining two interesting ideas that apply these concepts from physics to understand brain activity and structure. The first of these will be Einstein's theory of Brownian motion, and the other is a new concept called correlative spacetime. Einstein's 1905 work on Brownian motion helped explain how small particles in a liquid move randomly. When observed over a long time and many particles, this random motion of small particles leads to a predictable spread called diffusion. Diffusion here describes how particles move from areas of high concentration to areas of low concentration due to random motion. Einstein's approach effectively brought the microscopic, small-scale world into the macroscopic, large-scale world by connecting small, random molecular motions to larger, observable effects. In neuroscience, such diffusion principles can help understand how substances or signals spread between cells or networks in the brain. The second concept, connectomy spacetime, uses ideas from relativity to model the brain's network or connectomy. In relativity, the concept of spacetime combines time and space into a single structure, and the separation between 2 points in spacetime is called an interval. This interval depends on both the physical distance between the 2 points and the time it takes to travel between them. In this brain model, the interval between two neural events, such as signals between two neurons, depends on both the physical distance between the neurons and the time it takes for the signal to travel. Instead of the speed of light, this model uses the speed of neural signals, which is variable and much slower. The idea is that signals in the brain travel along the most efficient paths, which are affected by the structure of the brain, similar to how objects move along specific paths, or geodesics, in spacetime. This concept goes a step further by suggesting that intense brain activity can curve the brain's network, similar to how mass and energy curve spacetime in general relativity. Large objects, such as stars or planets, can curve space-time around them and affect how other objects move. The idea here is that high levels of brain activity can change the network pathways, making some connections in the brain more direct or accessible, similar to creating shortcuts in neural pathways. This analogy, while creative, faces several challenges. Unlike space-time, which is affected by gravity, the structure of the brain is not rigidly shaped by neural activity. Neural connections are adaptable and change with experience, making the idea of a fixed curvature more complex and dynamic. Neural signaling speed, unlike the constant speed of light in physical space-time, varies greatly and depends on specific biological characteristics. This variability makes it difficult to define a coherent neural space-time. Measuring such curvature in the brain is difficult. Techniques from neuroscience, such as network analysis, can help approximate these ideas, but applying precise physical concepts, such as curvature, remains speculative. Introduces two advanced concepts that apply theoretical physics to understanding consciousness and brain imaging: emergent 5D space-time and the holographic principle, and applications of diffusion theory to medical imaging. Let us now examine these ideas, consider their foundations, and discuss their potential implications. The concept of the ADS-CFT correspondence serves as a foundational idea in this chapter. Originally developed in string theory, the ADS-CFT correspondence suggests that a theory defined in a higher dimensional bulk space, such as a 5-dimensional anti-de Sitter or ADIS space, can be fully described by a lower dimensional boundary theory. This relationship is also called the holographic principle, where the higher dimensional space can be thought of as the projection of information onto a lower dimensional surface. Here, we can propose that consciousness may function as a holographic projection into the fifth dimension, emerging from 4D neural activity. The fifth dimension, labeled in this framework, could represent a different level of reality than the observable physical connection in 4D space-time, and could potentially serve as an interface or projection plane for consciousness. In this setup, consciousness would not be merely an epiphenomenon of neural activity. Instead, it could arise from complex interactions between the physical, 4-dimensional, and a higher-order 5th-dimensional realm. This fifth dimension could capture patterns or phenomena that our ordinary 4D vision cannot capture, effectively functioning as an extra degree of freedom that allows conscious experience to emerge. In physical theories, additional dimensions, beyond the 4 known space-times, are often proposed to unify different forces or explain quantum behavior. However, proposing a fifth dimension specifically associated with consciousness is speculative and has no empirical basis. The challenge is to define what properties this fifth dimension would have and how it would causally interact with the 4D connection. The holographic principle has been tested in simplified models of quantum gravity and black hole thermodynamics, but extending it to neuroscience and subjective experience is conceptually challenging. Designing experiments or observations that could detect holographic consciousness remains an open question. If consciousness could indeed be represented as a projection into a fifth-dimensional space, this idea would have profound implications for both physics and philosophy. It could imply that the physical structure of the brain serves as a boundary surface that encodes information in a way that manifests as consciousness in a higher-order space. This proposal has parallels with discussions in quantum mechanics about the role of the observer and the potential non-local nature of consciousness. We will now examine the application of Einstein's diffusion theory to advanced medical imaging techniques, particularly diffusion MRI and IVIM, intravoxel incoherent motion imaging. Einstein's diffusion framework describes how particles move randomly in a medium, and when applied to MRI, it helps map the microscopic motion of water molecules within tissues. In diffusion MRI, the primary measure is the diffusion coefficient, which characterizes how freely water molecules can move within tissue structures. This motion is affected by cellular boundaries, fiber orientations, and other microstructural features, making diffusion MRI a valuable tool for non-invasively inferring tissue architecture. Diffusion coefficients derived in MRI are mathematically expressed with imaging parameters that control sensitivity to diffusion. For example, higher coefficient values in MRI increase sensitivity to slower-moving water molecules, allowing for detailed resolution of smaller structural details within tissue. This method is particularly useful in neuroscience for mapping the brain's white matter tracts, because water diffusion along nerve fibers reveals the orientation and connectivity of these fibers. Intravoxel incoherent motion, IVIM, expands on standard diffusion MRI by accounting for two types of motion: the slow diffusion of water within tissue cells and the faster movement of blood within capillaries. By separating these two components, IVIM provides insight into both tissue microstructure and perfusion, blood flow within capillaries, which is particularly useful in cancer diagnosis and other areas of medical imaging. By applying Einstein's diffusion equations to these measurements, MRI practitioners can create high-resolution maps of tissue structure and blood flow, help detect abnormalities such as tumors or lesions, and improve our understanding of brain connectivity and function. While these concepts remain separate fields, on the one hand highly theoretical physics, on the other applied medical imaging, if they are linked together, they could influence new methods for mapping or interpreting brain activity in medical imaging, if higher dimensional theories of consciousness can be formalized and experimentally supported. While the five-dimensional approach to consciousness remains at the margins of speculative theoretical physics, the application of diffusion theory to medical imaging demonstrates the real-world utility of physics in understanding biological complexity. Both fields emphasize the potential for interdisciplinary frameworks that bridge physics, neuroscience, and medicine, but each requires different standards of evidence, formalism, and interpretation. A revised mathematical model is implemented that reformulates action potential conduction as a constrained relativistic process. Through a causal structure in the connectivity space-time, action potential trajectories, or brain lines, are confined within a relative cone-shaped volume, paralleling Minkowski's conceptualization of space-time. Action potential propagation along neuron axons within the brain connectivity space can be conceptualized as a kind of random walk. However, unlike standard random diffusion, this propagation must obey a speed constraint imposed by the axonal conduction velocity limit of approximately 100 meters per second, a critical deviation from the boundless speeds assumed in classical diffusion theory. Here, it is important to reformulate the diffusion process to meet this constraint and define a four-dimensional causal connectivity space-time that integrates both spatial and temporal dependencies in a relatively structured framework. A novel approach to modeling neural signal propagation is available by applying concepts from relativity within a framework that includes constraints on signal speed. This framework includes three key components: diffusion with relativistic constraints, connectome space-time, and brain lines with causal volumes. These ideas represent a bold attempt to combine neural activity modeling with relativistic principles, providing a structured way to understand signal flow in the brain. In traditional diffusion models, as originally described by Einstein, there is no speed limit on particle motion. The classical diffusion equation describes how particles propagate through time without any constraints on how fast they can move, resulting in a model in which the theoretical diffusion rate can be infinite. While this assumption is valid for microscopic particles, where random motion predominates, it does not match the biological reality of neural signaling. In the context of the brain, signal propagation along axons is limited by a finite speed of action, potentials of about 100 meters per second. This speed constraint is critical because neural signals are not instantaneous. They require time to travel through the brain's network and establish temporal relationships that affect cognition and perception. To accurately model neural propagation, a relative adjustment that respects this propagation limit is required. Including a finite signal speed transforms the propagation equation into a constrained model. This adjustment mirrors approaches in physics where models are adapted to account for speed limits, such as the speed of light in relativity. By introducing a propagation limit, the framework shifts from traditional propagation to what can be viewed as a relative model of neural propagation that accommodates both the propagation of signals and the inherent delay imposed by finite conduction speeds. To further expand the concept, we introduce the idea of a connection spacetime that contains a causal structure similar to the spacetime framework in special relativity. In this connection spacetime, the interval between two neural events depends on both the physical distance separating them and the time it takes for the signals to travel at their maximum propagation speed. In this model, a spacetime interval defines which neural events can causally affect each other. Events separated by more than the allowed spacetime interval will be causally unconnected. This structure introduces functional distances within the brain that are governed by both spatial separation and temporal delay. These functional distances can be thought of as constraints on which areas of the brain can interact within certain time frames, given the speed of signal propagation. In short, this structure implies that signals from a neural event can only affect nearby regions within a given causal volume. This approach to defining intervals in connectivity spacetime borrows from the concept of the light cone in relativity, where information can travel only within limits set by the speed of light. Similarly, the brain's connectivity would have a finite cone of influence, where neurons interact only within reachable distances in both space and time, ensuring that no signal can propagate faster than the axon speed limit. This conceptually elegant framework is not only physically plausible, but also offers a way to model brain connectivity that respects the temporal constraints of signal transmission. At this point, let's introduce brain lines, which represent the paths that action potentials follow as they propagate. In connectivity spacetime, brain lines define the trajectories of successive neural activations, forming paths that follow causal pathways. These brain lines fit into a relative cone structure that confines the influence of each neural event to a specific causal volume. In physical terms, each neural event generates a causal volume that encompasses the range of points that it can affect, limited by the maximum propagation speed of action potentials. This causal volume creates a double cone structure, an expanding region that extends back and forth in time from each event. The forward cone represents the range of events that a given neural activation can affect in the future, while the backward cone describes the region that it could affect in the past. Inspired by relativity, the double cone concept represents both past and future interactions constrained by propagation boundaries. In the brain, this means that each neural activation is confined to a temporally and spatially defined volume of influence, and only neurons within this causal volume can meaningfully interact. This constraint introduces a level of causality into neural processing, in which interactions are limited by both spatial proximity and temporal accessibility. In neuroscience, most connectivity models consider anatomical distance or functional correlations. Adding a temporal dimension in which connections are constrained by signal propagation times reflects a more realistic biological perspective. This could potentially improve our understanding of phenomena such as delay-based information processing or oscillatory coordination between brain regions. This adaptation is a theoretically robust model because it takes into account both physical constraints, relativity principles, and biological constraints, axon conduction velocities. Despite its conceptual elegance, implementing this model requires precise definitions of brain line trajectories and causal volumes, which can be computationally intensive. Practical applications in neuroscience could include simulations or graph-based approaches that map the brain's connectivity patterns under these constraints, allowing for new ways to visualize and analyze information flow. This model may be particularly useful for understanding brain coordination across regions. For example, it could provide insights into how large-scale neural oscillations synchronize distant areas across functional boundaries. This approach is not just a theoretical exercise. It has practical implications for how we can model, simulate, and understand brain activity. By incorporating temporal constraints, this framework emphasizes the importance of timing in brain connectivity, suggesting that functional relationships are as much about where signals originate as when they interact. It suggests a future direction in which neuroscience can exploit the principles of relativity, offering a path toward more realistic and temporally dynamic models of brain function. We will introduce the idea of causal independence in the brain by examining the relationships between neural events, such as activations of specific neurons or brain regions, within a single time slice. In this framework, neural events are causally independent if they lie outside each other's causal cones. This means that, at a given moment, two neural events that cannot affect each other within the limit of their propagation speed are effectively isolated. This concept leads to a flat connection space-time, similar to Minkowski space-time in special relativity, in which all events share a common temporal axis. In Minkowski space-time, only events within each other's light cones can have a causal connection, while events outside each other's cones are causally unconnected. Similarly, in a flat connection space-time, only events within each other's causal cones, limited by the maximum neural propagation speed, can interact with or affect each other. This idea of causal independence also suggests a parallel to quantum fluctuations in flat spacetime. In quantum field theory, vacuum fluctuations occur spontaneously and represent instantaneous changes in energy or particle fields, even in the absence of external forces. Similarly, in a flat connection spacetime, neural nodes can activate independently of each other in response to external or environmental stimuli. This model suggests that independent neural activations, or fluctuations, can arise spontaneously as isolated responses to environmental triggers, much like quantum fluctuations. Here, the structure of the brain allows for both independent and coordinated activations, depending on whether the events are within each other's causal volumes. By framing neural events as causally independent as long as they are not within each other's causal cones, this model emphasizes that a significant portion of brain activity can be locally isolated in time. In other words, the brain is not a completely synchronized network. Instead, it consists of multiple, functionally isolated areas that synchronize only when signal timing allows causal interactions. The analogy to quantum fluctuations is intriguing because it suggests that the brain may exhibit a degree of spontaneous, indeterminate behavior similar to fluctuations in the quantum field. This perspective suggests that neural activation patterns can sometimes be unpredictable and that certain events can occur independently of others. This unpredictability may play a role in spontaneous thought, creativity, or responses to unexpected stimuli. Unlike the particle fields of quantum mechanics, neural events are grounded in biological processes that have identifiable, if complex, causes. Demonstrating true spontaneous independence in neural activity requires isolating neural events that do not respond solely to local or global brain states, a difficult experimental challenge. Causal volume constraints in connectome space-time offer a new framework for understanding certain brain disorders, particularly by focusing on how abnormal neural pathways can disrupt typical causal relationships. For example, in schizophrenia, altered neural connectivity or altered rates of signal transmission may lead to asynchronous or even backward signal propagation. This means that neural signals travel along pathways outside the standard causal cone, creating unusual sequences in brain activity. In this model, such anomalous propagation can be conceptualized as non-standard brain lines that deviate from the typical forward-directed causal structure and result in a subjective experience of irregular or distorted temporal sequences. These atypical propagation patterns are similar to hypothetical suprathreshold or retrograde brain lines. Suprathreshold brain lines imply faster than typical conduction speeds, potentially allowing signals to reach areas outside the standard causal volume. This can create situations where signals arrive too early, disrupting the expected temporal order of neural events. Retrograde brain lines imply a backward flow, where signals propagate in the opposite direction, disrupting the temporal coherence of experiences. Such disruptions could theoretically contribute to phenomena such as disorganized perception, in which sequences of thought or sensory information do not align as expected. By framing atypical neural propagation in terms of connectomy space-time, this model may offer insights into the temporal disorganization often observed in schizophrenia. For example, the perception that events do not occur in order or the blending of past and present experiences. Abnormal causal structures in the brain may explain why patients sometimes experience fragmented or distorted realities. If specific disorders can be linked to measurable disruptions in neural propagation speed or connectivity patterns, this framework could open the door to new diagnostic approaches. For example, neuroimaging techniques that map propagation speeds reveal aberrations in patients with disorders such as schizophrenia. Therapeutically, interventions that stabilize or normalize signal timing could help realign these causal structures, potentially reducing symptoms related to temporal disorganization. This model could be extended beyond schizophrenia to other conditions that involve disrupted neural timing or connectivity. For example, in conditions such as epilepsy, atypical propagation pathways can lead to abnormal synchrony, creating bursts of activity that spread outside standard causal volumes. Disorders in which time perception is altered, such as certain mood or dissociative disorders, could also be re-examined through this lens, potentially revealing underlying disruptions in the brain's causal structure. By emphasizing that neural events are not globally connected, but instead interact within bounded causal volumes, it reinforces the notion that brain function is both temporally and spatially structured. In the context of disorders, this framework suggests that certain conditions can be reinterpreted as space-time disorders, in which disruptions in standard causal structure lead to symptoms such as disorganized perception or atypical thought sequences. This approach allows us to conceptualize and treat brain disorders in terms of connectivity and temporal coherence, rather than focusing solely on biochemical or structural abnormalities. By incorporating relative constraints into the diffusion model for neural action potentials, we create a structured four-dimensional connectivity space-time. This model not only resolves the compatibility problems of classical diffusion, but also provides a rigorous framework for understanding neural connectivity constrained by finite signal propagation speeds. The causal organization of brain lines in a connectivity space-time offers insights into normal and atypical neural processing, suggesting potential models for neurological disorders in which propagation constraints are altered. This approach highlights the deep intersection between neural activity and relativistic physics, opening avenues for further investigation of consciousness and neural dynamics in a structured space-time framework. By conceptualizing active neural nodes as masses that bend the connecting space-time, we bring the dynamics of brain connectivity into line with relativistic constraints, including effects similar to gravitational redshift and time dilation. This model offers a new interpretation of neural interactions by using an energy momentum framework to describe how action potentials propagate through a connectome shaped by neuronal activation patterns, thus combining neuroscience and GR principles. In this model, action potentials propagate along brain lines that are causally bounded paths within a curved connectome spacetime. By analogy with GR, the curvature of these brain lines around active nodes produces a so-called gravitational effect, similar to how masses bend physical space-time. In this video, we will also explore the mathematical foundations of this model, using the concept of energy density in active neurons to define a connectome-specific energy-momentum tensor. These foundations are the connectome space-time causal cones and the curved connectome space-time affected by neural activation. This framework draws heavily on the theory of relativity to describe neural interactions and suggests that brain connectivity can be understood through principles usually reserved for gravitational physics. In this section, we will introduce a connectome space-time framework that combines time along a vertical axis and two-dimensional space along the x and y axes to map neural connectivity. Here, causal cones are defined similarly to light cones in special relativity. These causal cones encapsulate the finite propagation boundaries of neural signals, determined by the maximum speed of action, potential conduction in neurons, approximately 100 meters per second. Causal cone boundaries represent the spatial and temporal boundaries within which any given neural event can affect others, and they define regions of causal connectivity. The model's causal cones, represented by a 45-degree structure around each neural event, define both forward and backward boundaries, representing the potential impact on future and past neural events within the neural network, respectively. Mathematically, the distance between two neural events, nodes, within this causal structure is defined by an equation that includes both spatial coordinates and temporal separation, and determines the maximum range for causality at any point. This adaptation of causal cones to neural activity has interesting consequences. Just as relativity places strict constraints on causal interactions between events, preventing information from traveling faster than light, the brain's space-time of connections constrains neural interactions to the finite speed of action potentials. This may help explain how the brain organizes signals across spatially distant regions in a temporally synchronous manner, despite the relatively slow speed of action potentials. Unlike photons in relativity, neural signals are electrochemical processes subject to biological variability, with propagation speeds influenced by factors such as axon diameter and myelination. This variability complicates the idea of fixed causal cones. Furthermore, modeling neural activity in terms of rigid causal boundaries can oversimplify the complex, distributed nature of brain connectivity, where indirect pathways and complex network dynamics play important roles. To introduce the idea of curved connectome spacetime, which proposes that active neural nodes create a form of curvature in the causal structure of the brain. Here, the curvature created by neural activation is analogous to the way large objects cause space-time curvature in general relativity, GR. According to this model, active neurons exert a pseudo-gravitational effect, changing the causal cone structure around them. This curvature is conceptualized through a metric tensor that describes the geometry of the connectome spacetime, reflecting the formalism of general relativity. Einstein field equations are adapted here to model the brain with a special focus on neural energy momentum, which depends on neural activity levels. The energy momentum tensor in this formulation reflects the energy in active neurons, reshaping the causal structure around them, and theoretically, changing the paths that neural signals can take. This model implies that the brain's connectivity is not static, but changes with neural activity. Regions of high activation can bend the connectome space-time, altering the signaling pathways around them. This curvature may provide a biological basis for how attention or focus affects neural processing, because regions of high activity attract more signaling traffic, similar to how gravity bends the paths of particles. Using the analogy of gravity, this model suggests the possibility that neural activity affects not only individual signaling pathways, but also the larger connectivity space. Such a model helps explain how brain states, e.g., resting and active, dynamically change connectivity patterns, potentially affecting the flow of information across large-scale neural networks. Curved connectome spacetime provides a framework for modeling neuroplasticity, where highly active neural regions influence the restructuring of surrounding connections over time. As areas of the brain become more active, they can attract and strengthen connections within causal cones, encouraging new pathways that reflect learned or repeated behaviors. In physical systems, curvature is measured by observing particle trajectories or the bending of light around large objects. Translating this to the brain, where signals are electrochemical and paths are not strictly defined by curvature, is a challenging step. While the metric tensor provides a mathematically precise way to express space-time curvature in physics, applying it to neural structures introduces uncertainties. Unlike physical mass, neural activation does not create a force that bends space. The curvature here is metaphorical and represents changes in connectivity patterns rather than physical distortions. In general relativity, space-time curvature is affected by fixed mass-energy distributions. But in the brain, neural activation is dynamic and transient, meaning that the connectivity space-time must be highly adaptable. This introduces a layer of complexity, as the causal structure is constantly reshaped in response to ongoing neural activity, a phenomenon that is difficult to model computationally accurately. Additionally, the introduction of curvature suggests that neural activation can actively reshape these causal structures, providing a dynamic model that can potentially capture aspects of attention, focus, and neuroplasticity. The architecture of connectivity suggests that it is not only a product of anatomical wiring, but also of functional, context-dependent parameters that reshape neural pathways in real time. In general relativity, mass and energy shape space-time through their effects on what is known as the energy-momentum tensor. In this model, active neural nodes, groups of neurons engaged in synaptic or electrical activity, are treated as localized energy sources. This energy density in these nodes is thought to form neural masses, altering the topology of the space-time of the connectome in ways that shape the paths followed by action potentials or neural signals. In this sense, neural activity not only transmits signals, but also actively shapes the space in which these signals propagate, creating a dynamic functional landscape within the brain. The concept of mass here is purely metaphorical, since it reinterprets the traditional mass in physics as a representation of neural energy without directly involving the physical mass of neurons. This approach simplifies the biochemical and cellular complexities of brain function, which can lead to significant distortions when modeling actual signaling and energy patterns in neural networks. The main difference between this model and general relativity lies in the speed at which signals propagate. In relativity, the speed of light is constant. However, in the brain's connectome, the speed of action potentials varies depending on factors such as axon length and diameter. In particular, axons with thicker myelin sheaths conduct signals faster due to a process called saltatory conduction, in which signals jump between points along the axon. The change in velocity is nearly linear, and longer axons generally conduct signals faster than shorter ones. This velocity dependence is analogous to gravitational redshift, in which action potential frequencies are reduced or redshifted as signals pass through areas with slower conduction velocities. In particular, shorter, slower axons experience longer recovery times between signals, resulting in reduced frequency transmission. The introduction of the redshift analogy is intriguing because it suggests that the brain may use a kind of information gradient similar to the gravitational potential as a functional organizing principle. The framework suggests that low-speed fibers with long refractory periods prioritize local, low-frequency signaling, while long-range fibers support fast, high-frequency communication between distant nerve nodes. This connectivity space-time model suggests that active neural regions act as gravitational sources, bending the paths that neural signals or brain lines can follow. Brain lines represent the trajectories of action potentials as they move within the constraints of this so-called gravitational field created by active neurons. This approach not only integrates neuroscience with the principles of relativity, but also provides a new way to interpret signal propagation and information flow in the brain. Unlike physical space-time, the connectivity space-time here is a theoretical construct intended to capture neural dynamics in a structured way. The model does not imply that the nerve nodes actually exert gravitational forces, but rather that the density of neural energy can functionally mimic the effects of curvature in signaling pathways. This opens up new research questions, particularly about how such curvature effects might affect cognitive processes, such as memory, attention, and problem-solving, or even neurological disorders in which signal flow is disrupted. By using structures inspired by general relativity, we gain new insight into how the brain organizes information in space and time, much as relativity organizes events in the physical universe. This theoretical combination of neuroscience and relativity, while still in its early stages, holds promise for advancing our understanding of the brain's complex structure and function. It could provide insights into disorders in which these connections are altered, as well as improve our understanding of how the brain balances efficient information processing with adaptability. Let's delve deeper into functional brain lines and causal volume constraints. In this theoretical framework, brain lines, which are paths along which action potentials travel, follow relativity constraints. Just as in relativity space-time, where events outside each other's light cones are causally unconnected, brain lines are constrained within causal cones based on an upper limit on the speed of signal propagation in the brain. This limit is here called the brain speed limit. A causal volume formula defines this limit and defines a spatial region around each neural event to which other neural events can be causally connected, as long as they fall within the speed limit of action potential propagation. This formulation is particularly important for long-range connections, where only certain regions within the connectome remain causally connected via brain lines. This adaptation of causal cones to the framework of the connectome is intriguing because it suggests that the brain may operate within a structured, speed-limited network of connections that influence not only signal timing, but also spatial relationships in neural processing. However, this concept of space-time inherent to the brain may be limited by the brain's inherent plasticity and variability, as neural pathways are continually reshaped by experience and learning, in contrast to the fixed speed-of-light constraint in relativity. General relativity explains how mass bends space-time and produces the gravitational effects we observe. In this model, the energy density in neural nodes serves as an equivalent source of curvature in the brain's connectome. Active neurons are proposed to act as mass analogs that influence brain wiring by concentrating energy in specific neural nodes. This analogy arises from the fact that active neurons undergo microscopic swelling due to water flow in dendritic spines. This results in a detectable, albeit small, increase in local mass. This increase in local energy density creates an effect similar to the mass-induced curvature in physical space-time, effectively guiding the paths of brain lines around active regions. This approach uses a modified Einstein field equation for the connectomy, where the constants are rescaled to fit neural dynamics rather than cosmic gravitational fields. In this setup, neural activation energy serves as the effective source of curvature that affects activation potential trajectories in much the same way that gravitational fields affect particle paths in relativistic space-time. The energy momentum tensor in this model represents the flow of neural activation energy, structuring the curvature geometry of the connectome and predicting how action potentials will propagate through this energy-structured space. The framework adapts the Einstein field equations to describe the neural landscape in which the flow of action potentials is shaped by the curvature generated by active neural nodes, effectively creating a unique neural gravity field. Unlike mass in physical space-time, neural energy is transient and fluctuates with dynamic brain states, meaning that the curvature in the connectomy is likely to be temporally variable. Moreover, while this analogy is compelling, it abstracts from other critical factors in neural communication, such as chemical gradients, synaptic plasticity, and network topology, which may play a role in shaping brain contours beyond just energy density. Future research could focus on incorporating more of these biological variables into the model, perhaps by treating curvature as a dynamic space affected by multiple parameters, not just energy density. Additionally, empirical studies using brain imaging techniques such as functional MRI or diffusion MRI could provide quantitative validation by helping to map how patterns of neural activity in this curvature correspond to predicted brain line pathways. By treating the connectome as a structured space-time in which neural nodes exert curvature through localized energy, this framework provides a new lens for understanding how the brain organizes information. This model suggests that brain connectivity can be structured around energy-based causal constraints, where the temporal and spatial relationships between neural events are dictated by both signal rate limits and local neural energy densities. The brain's connectivity landscape is not static. It evolves with each experience, meaning that any model must be highly adaptable to capture the fluidity of real neural interactions. However, the theory is spurring new approaches to understanding brain function, uniting neuroscience with physics, and opening doors to interdisciplinary research to investigate how fundamental physical principles may underlie the organization of cognition and consciousness. By reinterpreting GR principles for neural dynamics, this model proposes a curved connectivity spacetime in which action potentials follow brain pathways affected by the energy distribution of active nodes. This pseudo-gravitational framework reveals insights into the structured flow of neural information, showing how brain function, cognition, and even consciousness may be affected by relativity-like constraints on neural speeds. Thanks for listening and take care.